In this video, we're going to talk about how DNA replicates or copies itself, and how differences between DNA and RNA, and then how DNA makes proteins through a process called protein synthesis. So in this diagram, you can see here's a double strand of DNA, and it's got the double helix, and you can see it's sort of unzipping, and then there are new nucleotides being placed on each set. So this is an example of DNA replication. DNA replication is going to occur anytime a cell is about to divide. Remember, when we talked about the cell cycle, the first thing that has to happen is a cell has to copy its DNA, right? So we have two sister chromatids. So this is the process of that DNA copying itself. So let's look at this in a little bit more detail. So here we have our double-stranded DNA, the helix, the A's and T's and G's and C's paired up properly. So the first thing, if we want to copy this, we've got to unzip it. So there's an enzyme called helicase that's just going to come through the middle and unzip it. So it looks like that. Now we've got our bases exposed in the middle. And another enzyme called DNA polymerase is going to come through, and it's going to attach the appropriate complementary nucleotide. So if there's a guanine, it's going to attach another cytosine. If there's an adenine, it's going to attach a thymine and adenine, and so forth. So this is what's happening. The enzyme, you can't see the enzyme there, but it's attaching all of the proper complementary nucleotides. Remember, nucleotides just float around in the cytoplasm. They're just part of sort of the raw materials of the cytoplasm, so they can be obtained by these enzymes and attached on there. Okay, and then all we have to do is seal it up, and there's another enzyme called ligase, which basically will seal up and cement those nucleotides together. So you can see that it's not really the original DNA and then the copy. It's really each half of the DNA serves as a template. A template is like the sort of master copy upon which the new strand or the new helix is going to be synthesized. So it's that's how DNA is replicated. Now let's look at DNA and RNA. Now they're both nucleic acids. They both have a sugar, a phosphate, and 4 nitrogen bases, but they have three important structural differences that we'll need to talk about. So DNA stands for deoxyribonucleic acid. RNA stands for ribonucleic acid. So the first difference is fairly obvious. The sugar in DNA is called deoxyribose. The sugar in RNA is called ribose. And what does that mean? That means that there's one less hydroxyl group in DNA than there is in RNA, okay, in terms of the sugar. Then the second thing we have, in DNA we have A's, T's, G's, and C's. In RNA we have A's, G's, and C's, but instead of T we have uracil. Uracil replaces thymine. This is important when we get into protein synthesis and we transcribe DNA into RNA. We have to make sure we have a uracil in the RNA and not a thymine. Okay? And then the third structural difference between DNA and RNA is DNA is double-stranded. It's all the bases are sort of protected in that double helix. It's a nice sort of secure way to store genetic information. RNA is single-stranded, and we'll see that RNA basically just takes that message out to a ribosome to make protein. So it doesn't have to be sort of in this secure structure as much as DNA does. So it's single-stranded versus double-stranded. So those are the three structural differences between DNA and RNA. And now let's talk about this process of taking the recipe or the genetic information from DNA and then transcribing it into RNA and then making a protein, and that's called protein synthesis. Okay, so here again are the differences. Different sugar, uracil instead of thymine, double-stranded versus single-stranded. Here we go. So you can see DNA stays in the nucleus and it's double-stranded. It's going to get copied into a type of RNA called mRNA. Now M just stands for messenger RNA. This is the messenger. This is going to take that genetic instruction from the nucleus to the ribosome, okay, and there goes the mRNA out to the ribosome, and then it's going to get translated by another RNA called tRNA, and that tRNA is going to sort of read that genetic code and transcribe it into a sequence of amino acids, and the amino acids make up the protein, right? They're the monomer upon which proteins are based, okay? So we go from DNA to RNA to protein, and let's look at that in a little bit more detail. The first step here is called transcription. Trans means across. Scribe means to write, so we're rewriting the DNA code into an RNA code. So we're going to unzip that strand of the DNA called a gene that contains the recipe for that one protein. And we're going to, again, where we have a G, attach a C, and A, we pair U. We don't have thymine. Now we have uracil in RNA. Okay, T pairs with A, C pairs with G, A pairs with U, etc., etc., until we're done with that gene, and then we have that strand called messenger RNA. That's going to peel off, and it's going to go out to the ribosome with the instructions. Okay. When we get to the ribosome, the next step is called translation. You can think about translating as going from one language to another. And that's sort of what we're doing here. We're going from the language of nucleic acids, DNA and RNA, to the language of proteins, amino acid sequences. So we've got these RNA triplets. They're called codons, right? They were transcribed from the DNA triplets, 3 DNA bases were transcribed into these. They're going to pair with anticodons on different tRNA molecules. So if we have GGC, that'll pair with CCG, and that'll place this amino acid called glycine onto the polypeptide. If we have UUU, that pairs with AAA, and that one turns out sticks the phenylalanine on there. So basically what we're doing is we're going along reading all these codons. We're actually going in this direction, so this one would be the first one, second one, third one, and place the appropriate amino acid on there until we're done. The last codon on here is called a stop codon. That's going to say stop production. We're done. We're going to send that polypeptide to the Golgi to get packaged and wrapped up and shipped out to wherever it needs to go. So that's the steps involved in protein synthesis. The tRNA molecule that we looked at back here, it doesn't really look like these kind of cubes. It actually looks like a T. The 2 important parts of it you need to know are the amino acid attachment site. That's where it's going to grab and hold on to the amino acid so they can attach it to the polypeptide. And then the anticodon, that's important because that's going to pair with one specific codon. So again, if we have AAG, this would pair with a UUC on the mRNA, and only the UUC. So it's very specific here. This is only going to attach one particular amino acid based on the pairing of the anticodon with the codon. It's sort of like we're reading and interpreting this code, this message in the ribosome with tRNA. Okay, and so here's an example of tRNA translating the mRNA codon. So we have the AGG that pairs up with the UCC, and that attaches a specific amino acid right there. Okay, and that is protein synthesis. So the summary again, overall, we have in here in the nucleus, we unzip some of the DNA and we transcribe a certain sequence of it, the gene, the genetic instructions for one protein, into a molecule called mRNA. That mRNA then travels out to a ribosome where it is translated by these molecules called tRNA, and based on the pairing of the codons with the anticodons, tRNA molecules are going to attach a specific sequence of amino acids that get ultimately folded up into a protein. So that's how protein synthesis occurs, and that's how our cells make proteins. And proteins, again, do all the important stuff in our cells. So this is a really important process. And if we have mutations or mistakes in our DNA, we're going to have mistakes in the proteins, and we're going to have genetic diseases. And we'll study some of those genetic diseases this week in class. Okay, so that is it.